This is our spectrophotometer. We use it to calculate the concentrations of biomolecules in cells. Basically, it shines a stream of UV or visible light through a particular sample. That sample then absorbs some of that light. The spectrophotometer then calculates a ratio of the transmitted light through the sample to the incident light, and from there we get an absorbance value. From that absorbance value, we can actually calculate the concentration of the particular sample.
Here we have our spectrofluorophotometer. Similar to the spectrophotometer, it uses UV light to analyze biomolecules, except in this case, instead of measuring absorbance, the machine is able to measure fluorescence. However, this requires that the sample actually contain a fluorophore. Basically, what the machine does is it uses UV light to excite the molecules in the sample. These excited molecules then relax, and the relaxing causes the molecules to release their energy in the form of fluorescence.
This is our sonicator, which we use in a step in protein purification. It uses sound energy to lyse cells by disrupting the cellular membrane. Basically, how we do this is by placing the sample at the bottom of the microtip, and the microtip vibrates, and the vibration causes the cells to lyse, and we can then extract the contents of the cells.
Here we have our iActroScan TLC FID analyzer. Basically, this machine combines two techniques, thin layer chromatography and flame ionization detection, in order to separate and study various organic molecules. Firstly, what you do is you place your sample on what is called a chromorod. The chromorod then allows the samples to be separated out into its various organic molecules. This chromorod is then placed into what is called a hydrogen flame, and this flame allows the various organic molecules on the chromorod to be ionized and then quantitatively analyzed in order to give you the information you need about your sample.
This is our ultracentrifuge, which we use for sample preparation. First we put our samples in the rotor, and then the rotor into the centrifuge. The centrifuge then rotates the samples at extremely high speeds. This creates a centrifugal force, which then sediments the protein. Proteins will sediment according to the sedimentation principle, which means that they will be arranged according to the densities. This way we can separate cell fractions and biomolecules from one another.
So this is our DSC machine. DSC, or differential scanning calorimetry, is a technique that allows the measurement of temperature differences between a sample of interest and a reference. This machine measures the amount of energy, or heat, required to keep the sample and the reference at about the same temperature throughout the experiment. Along with other information about the thermal properties of the sample, the heat capacity and the temperature range of phase transitions can be determined. DSC has many useful applications. For example, it can be used for accurate determination of phase transitions, to test the calorimetric purity of pharmaceuticals, and to evaluate metal alloys. DSC is also a nice complement to other sample phase analysis techniques, such as deuterium NMR.
This is a reverse phase high pressure liquid chromatography column from Varian Incorporated. For short, we usually just call it HPLC. It's used in the final stage of protein purification in this lab. The column is packed with a non-polar stationary phase, and a polar mobile phase is pushed through the column under pressure. Polar molecules like proteins are then eluted faster than non-polar organic molecules, which stick to the stationary phase. A particular protein has a characteristic retention time that identifies it from other molecules in the element. These are collected in fractions, such that each fraction contains a different component of the analyte.
Boom, ba, da, boom, ba, da, boom, ba, da.